high or too low. Okay, so it has to be tightly controlled. While in the cell wall, in the vacuum, you can afford to have dramatic changes of the sugar levels. Yeah, like in watermelon, in tomato, it's sweet. Why it's sweet? Because the sugar is in the vacuum. It's not so much sugar levels in the cytosol. Okay, and consistent with that idea, indeed, the cytosolic invertase displayed very broad or constitutive expression patterns as compared to the acid invertase, across many species, both in Arabidopsis or tomato or maize and rice. Yeah, for example, here you can see the CO vacuole show some ice forms highly expressed in reproductive organs, while others mainly in vegetative organs. However, if you look at the CRN, cytosolic invertase, you can see they are very much constitutively expressed because every cell, regardless whether it's in what organs, all require some similar level of sugar homeostasis within the plant body. So to summarize, the key message here is that the cytosolic invertases are evolutionarily and functionally more stable as compared to the CO and vacuole invertases, probably indicating their roles in maintaining cytosolic sugar homeostasis for basic cellular function. Okay, so just to have a summary, I have show you or share with you some of our work that cell wall invertase and its inhibitors were evolved in seed plants starting from germspoon and then expanded in angiosperm, indicating their roles in sugar and fruit, in seed and fruit reproduction through controlling upper-plasmic phenomena unloading in case of, if it's through the cell wall matrix unloading, and through sugar signaling from the cell wall matrix when carbon is imported through PDs. Okay, the cytosolic invertases were involved in strong purifying selection, indicating their roles in cytosolic sugar homeostasis for fundamental energy metabolism and cellular function. This is why we do not see spontaneous cytosolic invertase mutants, because those mutations, those mutations, if ever occurred, would be most likely detrimental or fatal, lethal to the cell wall, cell function, or to the plant individuals or species. Okay, so I think I stop here. I hope I have some time to for everyone, for people to ask questions, and I acknowledge many people in my group. However, I don't want to read. It's not a complete list of the contributors, but those are some of our members taken last year. Thank you very much.
Thanks, Dr. Ruan, for a wonderful talk. I think we still have a few minutes for questions. So I think I saw like 2 questions pop off from the QA section. So the first question from this audience is that, is there any negative feedback mechanism as more hexose sugar are produced by enhanced activity of C-win? So that's the question. You can, Professor Ruan, you can also see the question from the QA section. If you.
Okay. I want to know, is there any negative feedback mechanism as more hexose sugars are produced by enhanced activity? I think that question refers to our tomato work, because we silence the inhibitor that allows the cell wall invertase activity to be increased. Yeah, and we do not see obvious under glasshouse situation, we did not observe any negative effect. However, we did see color change of the young fruit in tomato. As you might recall, in our 2018 plant physiology paper, that under heat stress, we found the transgenic tomato where CO immune test activity is increased. They should improve the fruit set and heat. Interestingly, we noticed that the young fruit appears to be less green, pale color. So indicate maybe the chlorophyll biosynthesis or fruit photosynthesis may be suppressed or inhibited by this extracellular, high sugar levels. Yeah, that I think is possible because some of you might recall that the 1990s when the concept of sugar signal evolved. Glucose can inhibit photosynthesis by using maize protoplast. Yeah, although there are some controversial arguments about that experiment, but I think I think is if you have high sugar in the surrounding cells, it tells the cells that okay, there's plenty carbon available. Why you need to photosynthesize? Yeah, so reduce the photosynthetic capacity by suppressing chlorophyll biosynthesis or by suppressing some of the light or dark reaction carbon cycle enzymes, ribisco, and so on. I think is quite possible. But anyway, so we this is only perhaps slightly negative effect we observed. So it makes the young fruit less green color. Okay.
So there's one more question. So I think you can say which mainly have functions on plant cell development is that have any relation with the fruit quality, especially for whether the fruit tastes sweeter or not.
Mm-hmm.
The sugar metabolism. Yeah.
Well, I think for fruit quality, I think sugar level is very important. Typically, people in China, we Chinese consumers prefer much sweeter, right? Fruit. So the sweetness comes from the sugar levels, mainly, the sugar accumulation in the vacuum, right? So from that perspective, we need to understand sugar transporters and sugar metabolic enzymes, and their regulations in the vacuum, particularly like those transporters on the tonoplast and the vacuolar immotes, so that collectively control the sugar level in the vacuum. And more importantly, or equally importantly, is that how the vacuolar sugar homeostasis or sugar levels is coupled or coordinated with that in the cytosol. Yeah, and because if the vacuum, the sugar in the vacuum, accumulated to a very high level, yeah, is there a possibility it might, change the tonoplast sugar transport system to mediate efflux from the vacuum to the cytosol, to feedback, regulate, the gene expression in the nucleus. Say, hey, I got enough sugar in the vacuum. Stop, expression, further. Whether that is the issue. If that is the issue, maybe you can gene editing those enzymes or those transporters to make the fruit or the organ to accumulate even more sugars, to make it even to make it super sweet, if you like. That's a speculation.
Okay. So personally, I have a couple questions. So I think you said increase the cell wall invertase expression level will increase the fruit set. So I'm wondering what is happening in the meristem. So will it like also increase the meristem cell numbers, or what happened? So can you please comment on that?
It means a meristem. You're talking about shoot apical meristem, like SAM.
Yeah. Not shoot, not shoot. I mean the fruits. You said the increased invertebrate level will increase the fruit set, like fruit, like seed number. So what happened in the meristem? So the cell number increased, or did you ever check that?
Probably I needed to clarify a bit. I guess during my talk, I basically fundamentally focus on two systems. One is use the tomato, fruit set, and later on I talk about Arabidopsis ovule formation. So the tomato fruit set, the cell wall immunity is highly expressed in the placenta of the fruit, connecting the young seed. The improved fruit set and heat stress is because the high cell wall immunity in the placenta region actually can block program cell death that occurred in the wild type and heat stress. So that explains. And and then in the Arabidopsis situation, we are asking the question whether ovule initiation from the placenta in that female reproductive organs, is also under control of this extracellular metabolic sugar signaling, even if the nutrient, the bulk of the carbon coming in through plasmatic stomata, through the simplex, not through the cell wall. So we certain cell wall immunity use artificial macroLNG and ovule specific promoter is called a stick. And so when we do that, it actually can block ovule initiation by significant by 50%. Our explanations, I didn't have time to talk about all the data set, but we have other evidence, including RNA sequencing and so on, is that if we silence the cell wall immunity in the placenta region specifically, it will affect expression of transcription factors, oxygen signaling genes. Many of those genes are known to be required for ovule initiation. Having said that, back to your question about the meristem, we did observe, in our in situ hybridizations that cell wall immunity gene mRNA is evidently detectable in the influence meristems. And that says it might control even earlier. The influence meristem also under sugar signaling. But how that is achieved, we don't know yet. That is a question maybe for future. But I want to say that in our studies, we did not find any effect of Trojano's sugar signal involvement. So you might, if you read the literatures of the last 5 years also, there's a concept of Trojano 6-phosphate as a sugar signal to control plant development. That might be the case in some scenarios, but certainly not in our case. So it's not involved in our ovule initiation control.
Okay. So one more quick question. So the sugar-related PCD, so is it also happening in the normal growth condition?
Sugar-related PCD under normal condition. Under normal condition, my answer is no or unlikely. Indeed, we have a paper in 2016 in developing cotton seed, for example. We use normal fertilized cotton seed or cotton flower as a positive control or as a negative control, biological negative control, and in comparison with our transgenic seed, where the immunity is silenced. So the seed from normal pollination fertilization, they do not show PCD and in 3 days after flowering. So the embryo, in the sperm, they are all good. However, in our transgenic plants, or if you emasculate your flower, stop pollination, you can artificially induce pollination. PCD.
Yeah.
So So you can use that as a control to test, whether your technology is reliable or not.
Okay. Thank you. So I think we're running out of time. If there is no more question from the audience, I think we're going to.
There are more questions.
Yeah, there's more questions.
Oh, you can see the chat QR.
I'm sorry. Yeah. Please go ahead and see the third question is that could you please kindly let us know your email address? So I think you can find the Professor Ruan's information from online. So I can answer that question. So the next one is
that's right. Yeah, I noticed one student asked me like, could you tell me your email address? Well, now it's 2020. You can easily find my email address just by searching, whatever database.
Yeah.
Okay.
So the next one. Professor Ruan, so before you answer the question, could you please read the question, because our audience cannot see the questions. Only us can see the question. So yeah.
Okay. Let's see. Sugar signal. How to determine difference when it also does work in the. I think there's one question asking me like how to differentiate cell wall invertase as a metabolic enzyme, to hydrolyze sucrose into glucose and fructose from its potential role in sugar signal. I think this is a very important and fundamental question applicable not only to invertase, but also any sugar transporters or other enzymes like hexose kinase, for example, trehalose phosphate synthesis, for example. It's very difficult. This is the challenge area. This is why I talked to my colleagues working on auxin, hormones. I think many of those guys, they understand the importance of sugar signaling, but I think the people feel it's very difficult to do. That requires quite a bit of brain work. Okay. So in our case, for example, our paper, I encourage you to read our paper published last week, about the cell wall invertase as a sugar, control OV formation through sugar signaling, but not delivery of carbon nutrient. So we provide some approach to how to dissect, the two potential functions. One way, there are several ways. One way, you can design experiments such that you manipulate the level of nutrient, to see if that trigger, or complement the phenotype or trigger the same response or not. If the system does not respond to changes of the nutrient availability, well, but yet, you show a clear phenotype. That says whatever phenotype you produce is independent of nutrient supply, right? So this is one way. Another way you can think about it, biochemically, perhaps you can mutate your protein such that you can abolish its catalytic function, to make this enzyme become enzymatically inactive. Then you put it into the system back, you put it back to the system and to see whether it can trigger some kind of phenotype. If yes, well, the phenotype is unlikely, is uncoupled from the metabolic effect, right? So those are like some of the broad, I can't go to the details, but those are some of the approaches I believe you can try. But people can, well, I encourage anyone, you just ask questions later. You can do that, right? Or you say cannot. They cannot.
Yeah.
Okay.
Yeah. Okay. So yeah, so you think you answer all the questions our audience have, or is there more?
I don't know. Well, you as a moderator, you can see more. You make a decision. I don't know what's I'm happy to to keep discussing with everyone.
Yeah. So there are, I think there are a few more questions. Probably you can input your answers by using the keyboard. So I think we're going to move to the next talk. So thanks, Professor Ruan, for your wonderful talk. Thanks for your time and patience with our audience. Thank you very much.
My pleasure. All the best. Goodbye,
Okay.
everyone.
Yeah. Thank you.
Okay. Bye.
Yeah. So we are going to move on to our next talk. Please let me introduce you our next speaker, Dr. Jialong Yao. Dr. Yao is a senior scientist from the New Zealand Institute for Plant and Food Research Limited. Dr. Yao received his bachelor degree in agricultural sciences from Huazhong Agricultural University in 1983 and a PhD in plant biology and biotechnology from Massey University in 1993. He was appointed as a scientist at the Horticulture and Food Research Institute of New Zealand Ltd in 1993, leading the apple transformation and fruit development research projects. Then he joined Genesis Research and Development Corporation Ltd as a senior staff scientist and a group leader in 2001 to study molecular mechanisms regulating wood fiber development. Since 2008, he has served as a senior scientist in plant and food research. His research has been focused on developing efficient genetic transformation systems for tree species and analyzing gene function using these transformation systems and modern genomic tools. His team have identified genes controlling fruit size, shape, seedlessness, and texture, and the genes controlling cadmium and the xylem development and wood quality. He recently started an adjunct position at the Zhejiang Zhengzhou Fruit Research Institute, CES, as a co-leader of the apple genetics and breeding group. So I think now I'm going to hand it over to Dr. Yao. Dr. Yao, the audience is all yours.
Okay. Good morning, good afternoon, depending on where you are. First of all, I would like to acknowledge the conference organizer to provide this platform to share our research. In particularly, I would like to thank you, Professor Guan Qinmei, to inviting me for a talk. I'm looking for my presentation. I need to, okay, share screen. Okay, so can you hear me all right? So today I would like to share our unpublished results from a project started about 3 years ago. So you can notice my title is slightly changed, just to include our most recent result on the transformation regeneration. So we're trying to improve hypertransformation use the BBM gene. So what's baby boom? So short for BBM. So it's transcription factor with 2 AP2 binding demands. So there's 2 AP2 demands. There's something in the middle called a link demand. It's N-terminus on the terminus. So it's a typical member of the AP2 family, basically. The gene has been shown to regulate plant embryo development as well as meristem activity. So the gene has been used to enhance somatic embryogenesis and also for shoot regeneration in tissue culture. So most of these reports are in model plant species like Rhabdopsis or tobacco, are also being applied in some crop species as well, such as maize. But there's very little really reports on woody perennial trees. So that's what I'm talking to you today, how to use this gene to enhance of regeneration transformation in apple trees. Okay, so just give you a very brief summary on apple transformation. So the transformation is genotype dependent. So some cultivars is transformable. So most of cultivars are not transformable. So the transformation is also operator dependent. This means someone can do it, someone can't. So even in the same laboratory. So also, let's say the small number of laboratories are being able to successfully produce hypertransformational plants. So the transformation efficiency is something around like 0 to 3%. So there's a variation between experiments. Sometimes you get a no transformant produced, sometimes you have a few. So it's just a recent review by our Japanese colleagues, so one of the Japanese group. They basically summarized the transformation efficiency on apple rootstock called JM2. So the transformation efficiency for this particular rootstock is about 0 to 3%. I think that's probably the same for other apple cultivars, like we work on raw Igala, we probably get a similar transformation efficiency. So it's really the improvement is required for its wide application of the transformation system, because sometimes I struggle to produce sufficient numbers of transition lines. I think that's not just a problem for apple, it's a problem for most of woody trees like pears, peaches. Peaches is almost impossible, or other trees like even oranges are really hard. So, okay, so the aim of this study is really trying to analyze the function of apple BBM genes, also trying to determine if baby boom can be used to enhance epigenomics on the social regeneration. So if it can, so then we're trying to identify baby boom transition lines for reuse in transformation experiment. So particularly for gene function testing. So first of all, we're trying to know how many baby boom genes in apple. So when we started the project, there's one apple reference genome published in 2010. So it's from the golden delicious cultivar. So from this reference genome, we actually identified 2 genes, named as BBM1 and BBM2. So in BBM2 actually has 2 sequences, but these 2 sequences are identical in sequence. Just one is a shorter version of another one. So essentially, it's 2 genes. So more recently, we have more reference genomes. So one is probably in 2017. So also from golden delicious, but it's from haploid golden delicious. From this reference genome, we can identify 1 gene. So this MD04G. So this is baby boom 2. Okay, so there's no baby boom 1 gene identified from this reference genome due to whatever reasons. But we identified 2 genes from the harmful reference genome. Also is a haploid reference genome, so 1 gene here and 1 gene here. We also identified 2 genes from our own reference genome unpublished yet. So from haploid Roigala. Okay, so basically there's 2 genes in apple. So the amino acid sequence is very similar between the 2 genes and between the 4 reference genomes. So the first gene is like an outer group. Okay, so the homology is already in the AP2 demands. So what other genes this homology from the N terminus all the way to the C terminus? Okay, so it's very conserved sequence. We would like to know the if the gene is expressed or where it's expressed. So first of all, we have a check of EST databases. We did identify a single EST clone and the sequence matched to BBM1. Okay, and this is the 80 sequence we identified because the genes are supposed to be expressed in marshmallow tissues, but all the libraries are made from fruit, flowers, and leaves, so there's no expression. So this single EST from a normalized library of many different types of tissues, basically. So then we check some of transcriptome data. So in my laboratory, most of transcriptome data has been made from leaf and flower tissues because my research is focused on fruit development. So I'm not sure you can see the graph here because it's sort of like under this things been covered. But anyway, I tell you, is the expression is in fruit core tissues. Okay, so not much in open flowers or fruit skin or fruit flesh. Okay, the expression in the fruit core at about 4 weeks after pollination. The fruit core contains the developing embryos. So that's possibly the location of expression of the gene. We really need to do more transcriptome analysis, use young embryos or use shoot tips as a material. Okay, so we then made 2 types of transformation vectors. Use BBM1 because we already identified BBM1 expression in our EST database. So one of the construct is fusing the BBM gene with the GR gene. So the GR BBM fusion protein supposed to be localized in the cytoplasm, but can be translocated to the nucleus after treatment of DEX. So dextromycin is steroid type of hormone, basically. So as I introduced earlier, so AP2 is a TF. So TF supposed to be functioning in nucleus. If the fusion protein in the cytoplasm, so it's not going to function. If you treat it with DEX, they will move to the nucleus and then will be function. So it's 2 contracts have been made. So let's just show you the structure of the fusion protein. So it's the baby boom coding sequence, and then the GR protein is fused to the C terminus. Okay, and then the whole cases coming into the PRX778 transformation construct. So the first thing we did was to test if the vectors work in plant. So we have done Nicotiana, Benzimiana transformation. Okay, so we produce a bunch of transgene lines. We testing the expression level of the transgene. So let's the control produced with the gas gene construct. So let's three lines with a baby boom construct. We have more lines, about 80 tasteless three. Now have many lines produced with the BBMGR construct. So the majority of transgene lines show off expression of baby boom gene. Okay, so that's good. Then we're trying to test if the DEX induction works or not. So based on the transgene line without DEX on the medium, right? So this one is with 5 micromolar of DEX on the medium, is sort of like an induction medium. So regeneration of shoes on a EMS medium with very low level of cytokinin BA. So there's a 0.1 milligram per liter of BA. So normally tobacco shoes regeneration we shall use 1 milligram per liter of BA. So here is 0.1. So we're trying to say the effects of induction. If you use a high concentration of BA, so just regenerate too many shoes, you can't count the numbers, right? So that's basically our construct works, the induction works in tobacco. So another thing I noticed is interesting is what the shoes regenerate, the shoes regeneration from the bottom size of the explant. So if the size touch to the medium, so maybe the translocation of the DEX is very difficult in the tissue. So the downside touch to the medium has absorbed the DEX. So that's the place of shoes reduction. Okay, so our contrast working fine. So now we start to doing Apple transformation. So we use also 3 construct, the baby boom construct, the baby boom GR construct, also plus the gas gene construct. So we produce about 27, 29, 10 lines for 3 different construct. Okay, so the first thing we did, we also check the expression of the transgene. So that's for the baby boom transgene lines. You can show that's the control. No expression in leaf tissue. Okay, the transgene lines, the majority of the lines show off expression of baby boom. Okay, so that's been done by digital PCR. So the relative expression level is the BBM transcripts on ratio to the actin gene transcripts, basically. Okay, so actin is the housekeeper gene expressed at a relatively higher level. So our transgene, which is about like 10% of the housekeeper gene. So that's the same. That's just the BBM GR, or that's the baby boom. That's the baby boom GR lines. Okay, that's just 2 different constructs. So we also show off expression on the control lines. Don't show any off expression. Simple. Okay, then we're trying to say if the DEX induction work or not working in Apple. So we have tested a number of transgene lines. There's no induction here. Show a slight increment of number of shoes per plate. So that's been done like on EMS medium plus 1 milligram per BA. Apple regeneration medium use much higher concentration of cytokinin. We have to use 5 milligram per liter of BA plus TD rate in a normal standard regeneration medium. Now we reduce them to 81 milligram per liter of BA. Okay, put 15 explant per plate. So that's about 2-fold in enhancement. It's not very much. But the sense is the induction doesn't work. There's no difference between plus or minus DEX. So that was the same. There's not other lines here. So the sense is the DEX induction doesn't work in Apple. Some of colleagues trying is before also not showing any induction. So because there's no we focus on what our future work will analyze on the BBM construct rather than the BBM GR construct. Okay. For the BBM construct, the first thing we notice change in phenotype is enhance of proliferation of shoes. So we're doing subculture of apple shoes, we subculture single shoes, like about 4 weeks later in the control will produce like 5 to 6 shoes. So you can see the graph here, so the average is about 5 shoes. So in the transition lines, okay, so half of transition lines also produce about 5, 6 shoes, but some transition lines produce like 10 to 15 shoes. Okay, so it's enhanced of shoes proliferation. Then we're trying to say if the enhanced of shoes regeneration. So this has been done similarly on images medium with no concentration of BA, 1 milligram per liter. So this control, this gas gene control. Okay, on this medium produce quite a large amount of colors, but not really 80 shoots there. So you can see from our baby boom transition lines, so these shoots are produced, okay? So it's clearly significant increase of shoots regeneration. Then we have a look at the standard medium, all right? So on the standard medium, which is 5 milligram per liter of BA plus 1 milligram per liter of IAA plus 1 milligram per liter of TDJ. So this is the gas control transition expand. You can see there's about regeneration here, and it's still quite a reasonable amount of shoes regeneration. But you can see the baby boom transition lines, there's heaps much more. So it's not bad. We just give up to counting the numbers, just impossible to counting the numbers. Okay, so just show the photogram here. So it's a great improvement on the shoes regeneration. Okay, the next thing we're trying to say is if the transition apples with overexpression of baby boom are phenotypically normal or is phenotypically abnormal. So we have to say the plant can grow relatively similar to the control, okay? But they do show some modified morphology, like smaller leaves, or the leaves about curly, or even the shoots about curly. So this is about 3 months after micrografting. So we produce tissue culture shoots, and then we graft onto the dwarf rootstocks morning line. So that's 3 months after grafting. So that's about 7 months after grafting. We already reached about like 2.5 meters, 3 meters tall, has produced more than 120 nodes. So hopefully they will flower. This plant will flower after the winter dormancy breaking. Okay, then we have looked at the leaf tissue in about more detail. So you can see the control leaves, and there's 2 different types of leaves from the BBM transition lines. One looks largely normal, another leaves like quite curly. So the curly leaves may be due to uneven cell division or cell expansion. So we did some section of the leaf. So on the control, you can see a normal upper epidermal cells, there's a parasite cell layers, there's spongy cells on the lower epidermal cells. Okay, so the leaf tissue structure is quite defined. Okay, the cell types is very easy to define. In the baby boom transition lines, you can say there's increase of cell lines in the parasite cell lines. Okay, you get about 3 cell lines rather than 1. Okay, so that's the same. But also they have more cells. The cells are much more closer. There's no intercellular spaces between the cells. Here you can see there's a lot of intercellular spaces, but there's another section like just parallel to the leaf. So there's a cross section of the leaf, there's a parallel section. So you can see here is a lot of space between the cells. So in this 2 graph, you don't see much space. So let's indicate of expression of baby booming apple enhance of cell division and increase cell numbers in leaf tissue, and this there may be increased cell numbers in the margins, which is about harder to observe or fruit. We don't know. So the plant is not flowering yet. Okay, so the most important thing is the next step to say if the baby boom transgenic lines can enhance of transformation. So we have to do a retransformation, and we have to use a different type of selection. So the plants previously produced with calamithin selection. So now we have to use the ALS gene and with herbicide selection. So a single amino acid change in ALS can force herbicide resistance. So the gene we call AAM, which is apple ALS mutants 1. Okay, so it's also under 35 years promoter. So this country has been published 2013 in Plancy report. Okay, so that's how the transformation looks like. So we test 2 lines plus the gas control. It's roughly about like 2 months after co-cultivation in darkness. That's why it's yellow, okay, the color. And you say the control lines produce colors on herbicide selection. So that's not really much of shoots or bud regeneration. But in the 2 transgenic lines, you can see there's bud come out quite a number of places, okay? So now we look at if these shoots are transgenic going to turn green or not. So the shoots regenerate in the darkness will be isolate, then put on the medium still with herbicides, then in the night, under night. Okay, so 2 weeks with lighting, you can see the putative transgenic lines green up, okay? The shoots multiplication continues. So there was 1 bud, now they multiply as a clump of bud. We count this as a 1 independent transgenic line, okay? So some of these lines are not transgenic, so they regenerate in the dark, and then put in the night, they're not green up, so they are escapes, basically, okay? So let's just control of the gas construct. So let's not from the retransformation, just a normal regeneration shows. Let's just to show our selection pressure is high enough because the control are yellowing out. The control plant from night to starting with, okay? They don't continue to multiply yet. So that means our selection pressure is good enough. So we have done a number of experiments. Most of the experiments still in progress. 82 experiments close to be finished. So our transformation efficiency for lesson line is about 30 or over 30% in 2 experiments. So we're waiting for more results to come out. So that's about what? 10%, tenfold, 10 times increment on transformation efficiency, which is quite good. Okay, just like summary. So apple has 2 baby boom genes. We choose to overexpress BBM1 in transgenic apple plant. We found the overexpression enhance shoot modification, enhance the shoot regeneration, change leaf shape and size in greenhouse, increase cell numbers in leaves. The most important is improve transformation efficiency. Okay, so we found the GR-DEX induction system did not work in apple. Also, it worked in tobacco. So, because now if we can produce sufficient number of plants in 1 or 2 transformation experiment, we think we can produce enough transgenic lines within 6 months ready for micrografting, because the regeneration will take like 2 to 4 months, 3-4 months, and then a couple of months multiply elongation or ready for micrografting. After micrografting in the past, we know we can flower the plant in 16 months. So that's basically, we probably have about 2-year turnaround from transformation to flowering. Okay, so we think possible future work. So we will screen more baby boom transition lines. So far, we already screened 2 lines. We really want to identify the ideal lines for use as starting material for retransformation. The lines has to be highly efficient, but has a minimum change on phenotype. Because we use a 35 years promoter, it's most likely going to change the phenotype. So that's a problem. We use the induction system, but the induction system doesn't work. So we're probably trying to understand about mechanism how baby boom enhance cell regeneration, doing about more microbiology to identify the partners working together with baby boom, things like this. We're probably trying to apply the concept in other tree crops. The one we're trying to do now is pair transformation, so could be used for other trees as well. Okay, so I would like to acknowledge my colleagues. So the work has been done in the last 3 years, number of people involved. Most recently, Jialong joined the project. He from Huazhong Agricultural University. His visa supported by China Scholarship Council. The project is supported by New Zealand Ministry of Business, Innovation and Employment, MBIE. So Andrew has involved from beginning on design the concept. So Smarty has produced the apple transition plant, the baby boom transition plant. And Luke has helped on digital PCR. So when they help us maintain or bulk up the transition plant. We also would like to thank to our greenhouse team to maintain our plants in the greenhouse. So I would like to thank the audience for your attention, and I would like to answer any questions.
Dr. Yao, thanks for the great talk. I think it's very informative. So I can see one question from the QA section. You can also see the question. The question is, DX induction system does not work in Apple. Is it because that there is a leaking of the BBM1 GR system and even small amount of BBM entering the nucleus is enough to work in regeneration? Or the function of BBM in regeneration is not the TF?
Yeah, I hard heard your question, but I'm looking at it. So DXY induction system does not work in Apple. It's because that there is a leaking of BBM GR systems, or even small amount of BBM into the nucleus enough to work in regeneration. Okay, the sense is there's a leaking, okay, for sure, but the leaking is not enough. So that's why they have a slight enhancement on regeneration even without the induction. So that means there's a leaking there. But with the induction, there's no fork increment. So the induction does not work. There's maybe some components are missing in Apple for the whole system to work in. So we are not in the era of making the system working. I mean, this by itself is another study to develop inducible systems in Apple or in tree species.
Okay, so I think there are 2 more questions. The question from Chen is a great talk. How would you do the transformation in pair? I think he might want to ask that, will the system also work in pair?
I tell you how to do in pair transformation is similar as Apple transformation. We use the same protocols for both. They works.
Okay. Another question is, Dr. Yao, thank you for the speech. I want to ask one question. How did you think about to use DEG into apples? Sorry.
You mean differential expression genes? What DEG you mean here?
Yeah, I don't know.
I'm not sure it's DEG or DEX, maybe DEX.
Yeah,
so,
yeah, maybe.
Yeah, I think it's DEX. We just put these things in the medium, okay? So it's everyone does.
Okay. So there's another question. BBM transgenic lines show the more dense deep cell numbers. Do you observe any enhanced photosynthesis and enhanced growth your transgenic lines?
We have not made the photosynthesis in this ginger plant. I mean, we could do, yes, good suggestion. Enhance of growth, because our greenhouse space is very limited. We can't have enough copies for each line to really phenotype in the growth rate, because you can't compare one plant with another plant. You have to have multiple plants of each transgenic line, so then you can have a decent comparison. But so far, I would say it's similar as a control, maybe slightly fast, grow faster, but it's not really clear.
Okay, so I have a question. So I think in other species, if you use BBM to increase the transformation efficiency, sometimes you might say reduce the fertility in the transgenic lines. So I think you said that your apple tree is not flowering yet. So do you worry about the fertility of the transgenic plants?
Maybe, but we have produced the tobacco plant. The tobacco plant is normal, it's fertile. So
it's fertile.
Yeah, it's no problem in tobacco plant. For the apples, we don't know yet.
Okay. So there's another question. So what kind of promoter will be ideal for baby bloom, like for the system? So I think
the inducible promoter is ideal, but there's no ideal inducible promoters.
Yeah. Okay, I see. I think there are more questions coming from the QA section. So let's say, Dr. Yao, have you ever checked the other gene expressions such as cell division related genes?
That's what we plan to do. We're going to do a transcriptome analysis of the transgenic lines to see the change of gene expression. Yes, this is plan. We'll do in the next few months.
Okay. So here's another question also about the pitch transformation. How to transform pitch?
Sorry, I have never done pitch transformation.
Okay.
I know it's really hard, pitch transformation, but personally, I have not done yet.
Here's another question. BBM affects embryo development, and what happened in the BBM transgenic apples? I think you already answered that question because your apple.
Yeah, we don't have fruiting yet. I guess you maybe has, I don't know, multiple embryos. But depends. They're not necessarily going to change the embryo. Each species is different. So in the model plant, they show enhanced embryogenesis, but in our case, there's no somatic embryo formation in our tissue culture systems.
Okay. The other question from Chen, Professor, I saw that your regenerated apple seedlings bloomed early and bloomed in 16 months. Have you done any further research on this early flowering? What is the mechanism?
For normal Roigala plants, because the starting material we used that was already adult material, right? They already passed the juvenile phase. Okay, that's ADAS material can flow when the trace reach about 120 nodes. Okay, after reach 120 nodes, they normally flow. Okay, this will be required few months, 9-10 months growth, and then has a winter dormant, and then next spring they'll flow. Yeah. So I have published a paper a long time ago in Plant Cell Report to show the transgenic plant can flow in 60 months. It's probably in, I don't know, about 1999 in Plant Cell Report. Search for it.
Okay. One more question. This overexpression system, whether can also use in mulberry?
Maybe you already know after you try it.
Okay. So another question is it possible to use a cytokinin inducer promoter to drive BBM for transformation?
Yes, I think so, if you have one, but you also have to demonstrate the cytokinin inducer promoters working in apple. I mean, depends on what the concentration of cytokinin is required. Yeah. Because cytokinin itself will enhance of regeneration.
Okay. So one more question is, Dr. Yao, do you think BBM will work in citrus?
Citrus, yes, most likely. You just need a patient to taste.
Okay. So another question is, it's all about different species. How about grape wine? Whether have done the same experiment in grape?
Yeah, we don't doing grape transformation. I know other laboratories does. Grape transformation, they do use somatic immunogenesis system. So yeah, may working well, because the baby boom is really supposed to enhance of somatic immunogenesis.
Okay. So here comes a question that myself is also very interested in. Have you ever combined BBM with WuXi or STM in your experiment?
No. So from the result we have so far, I don't think we need to combine with WuXi or STM, because the BBM by itself already has quite a dramatic improvement. So if you get more genes in, your phenotype is going to change too much. So we don't want a growth change on plant phenotype. Okay.
Yeah. So Dr. Yao, have you ever tried the BBM from other species instead of the apple BBM? Like
no, I know there's BBM gene control from a range of species. You maybe can use gene from different species. They may work, may not, because they do need the partner to working together. If the BBM gene from different species is not compatible with the partners in apples or in your own species, so may not work. So there's a chance may work or may not work. So you just have to try and change different genes if one gene doesn't work.
Okay, yeah. So for example, in some species, there are more than one copy of like a BBM gene, like in your apple, and like in other species. Do you suggest us to try every one of them, or just pick a few that most related to the
yeah, that's about lucky. Yeah, I mean, sometimes has more genes which you want to try. I think you will try the protein making most sense. If the protein has been changed, you maybe don't touch it. Also look at the expression pattern of the gene. Say if this gene expressed at the right place. So if they have quite a different expression pattern, they may have slightly different function. Yeah.
Okay, I see. So one more question from the QA. Dr. Yao, what is the routine medium you used to plant tissue culture, apple seasoning?
Okay. So first of all, we don't doing apple routine anymore. As you know, we're doing micrografting, right? Okay. So, but we used to doing apple routing, okay, before, like long time ago. I'm doing apple transformation from 1992, okay? So let's 28 years ago. So at this time we're doing routing. So our routing medium just half years plus about like 1 milligram per liter of IBA. So you can look at my old publications, you shall find the routing medium. So was an implant report published in about 1995.
Okay. So another question is, DNA methylation affects shoot regeneration, whether there are some relationship between BBM expression and DNA methylation or not?
The answer for this is I don't know, but my guess is that not, because baby bone not going to change DNA methylation. But tissue culture itself may change DNA methylation, but most of the time is the methylate DNA has no level gene expression, or we making regeneration will be even harder, I think.
Okay, I think that's probably our last question. So, Dr. Yao, why choose BBM? Are there any other genes have similar effects?
And yes, there's other genes has similar effects. As the previous Christian already said, there's a VOCR, there's STM, there's the KN1, there's
Okay.
yeah, there's a number of other transcription factors, yes.
Okay.
But we choose BB bomb is just like, seems like this one is working in more species than other genes, yeah.
Okay. So I think time is up. We have to stop here. Dr. Yao, thank you for your great talk, and thanks for your time and patience with our audience. Thank you very much. So now
Thank you.
we're
Okay.
we're going to move on to the next talk. So our next speaker is Dr. Yongling Wang from College of Horticulture, China Agricultural University. And she's a postdoc in Professor Tianzhong Li's lab. And their research interest is the movement of macromolecules and the mechanisms of RNP complex long distance trafficking in plants. And I think I will hand it over to Dr. Wang. Dr. Wang is all yours.
Okay, thank you. Okay. Good morning, everyone. Thanks for the moderator's introduction, and thanks for the two professors' wonderful presentation. It's my honor to have the chance to give a presentation here. My name is Yongling Wang, a postdoctor of Professor Tianzhong Li, come from China Agricultural University. Today, I present Professor Li to talk about the topic cheapening CCT, assemble RNP complex, and facilitate mRNA long-distance trafficking in foam. First, we will introduce it from the application of grafting in a horticulture group. As we all know, grafting is the main way of vegetative propagation in horticulture crops, and especially in woody fruit trees. In China, the main rootstock of pear is duli, doli, and so on, and the saran, yali, jingbaili for the cultivator graft on the rootstock. And through grafting, the phloem and the xylem are connected together between rootstock and saran to communicate the photosystem humor macromolecular in the phloem and the water nutrients in the xylem. In the production, we found that the saran shows different characteristics when grafted onto different rootstocks. The rootstocks can affect the development of the saran, for example, fruit quality, and increase the fruit yield. Also, it can affect the early flowering and the growth. It can make the plant develop and so on. Similarly, the siren can also have an effect on the rootstock. For example, to absorb more nutrients, tolerate the abonic stress, and increase the root vigor. So what caused this phenomenon? In the past, studies on the interaction between the rootstock and the sun mainly focused on 3 points, including anatomical mechanism, nutrient and transportation, and endogenous humor. With the discovery of macromolecules found in recent years, RNA signaling molecules became the focus of attention. In recent years, some plant endogenous mRNA was identified as a long-distance trafficking signal in hair graft system, and the trafficking of these mRNAs induce the phenotype changes in different tissues. For example, the leaf, the shoot plaques, the flower, and in the rootstock, the tuber, the stolon tip, and the root, and so on. Nevertheless, the function of most full mobile mRNA remain largely unknown. Well, to avoid the degradation during long-distance trafficking through the form, the mobile mRNAs must bond by RNA binding proteins to form an RNP complex. It enters shaft tube from the kidney cell by passing through the plasmodesmata, transport from the source tissue to seek in the form sac, and then unloaded in the target cell for the further RNA degradation and so on. For example, protein expression and further translation or occur to regulate plant growth and development. While in this process, the plasmodesmata only permit the molecules that is lower than the exclusion limit, generally between 10 to 60 kb. However, the RNP complex formed by RNA and protein it is much larger than the sense of exclusion limit. How these RNAs and the proteins are assembled and pass through the plasmodesmata for long-distance trafficking in form. These are all unclear. Here, we would like to focus on some questions. How to identify the mobile mRNA in woody plants. The second one, what is the function of mobile mRNAs? And finally, what is the mechanism of mRNA trafficking? Now, the first one, how to identify the mobile mRNAs in woody plants. In our study, 2 species of pear, duli and yali, is used as the metals. Duli is widely used as a rootstock in north of China. Yali is a cultivator grafted on it. Here, we use the tissue cultured yali as a sign grafted on yali itself or on duli. 3 weeks after grafting, total RNA was extracted from the cell form of the yali, and all samples was sent to the cabinet for further RNA sequences analysis. We assume that when the transcript can transport from the rootstock to the cell, the relative abundance of the transcript can upregulate. Briefly, we think that some upregulator transcripts are more likely as the mobile mRNAs. In our RNA sequence data, we identify about 1,030 is upregulated and 380 downregulated genes. Furthermore, about the specific sequence, 8% of the upregulated transcripts contain TLS, it means tRNA motifs, and about 5% contains CU domains. These are thought to be a structural feature of mobile MRIs. Once these potential mobile MRIs have been discovered, how to further identify the mobile MRIs? So first, we found that most MRIs in rootstock and serum have several SNP losses between two species. So CAPS RT-PCR experiment was performed to strongly demonstrate the mobility using Yali and Duli micrographed combination. Consequently, the RT-PCR product applied from rootstock cDNA and the serum cDNA could be cleaved into one or two fragments by the restriction endonuclease from two species. If the transcript can move through the form, we found that there may be two species of the fragment. So here we found that the serum has the fragment in the Yali and the fragment in the Duli. So it means that the transcripts can move long distance from the stock to the serum. Now, the second method is suitable for the MRIs, which has tissue specific. That is girding. We removed the phloem of Duli lateral branch at 2 points, and so the branch are precipitation into 3 parts. An interesting phenomenon was observed that the MRI was detected in the first part and in the second part, but it was disappeared in the third part at 2 days. And as time goes by, we found that at 32 days after girding, we found that the transcript appear in the third part again. And this means that the MRIs can transport from the stem to the trunk through the form, and the girding inhibit its mobile. And similarly, we also use this method to identify the transcripts move in the trunk. We also girding the form with or without lateral branch, and we found that the transcript in the trunk, we can detect the MRIs, and we can detect the MRIs when it is with lateral branch, but we cannot detect the transcripts when it is without lateral branch. So it means that the transcript with tissue specific, that it is pressed in the stem tip, it can go through the form and to the trunk. Now, the second question, what is the function of the mobile MRIs? There are few studies on the function of the mobile MRIs in fruit trees. In previous study, we found that ATGAI could cause the development of apple trees. Recently, we found that PBDRM can improve the drought tolerance of Duli. However, these are an important aspect on drought tolerance on mobile mRNA research. It will be an important application of mRNA long-distance trafficking in fruit trees in the future. Now, the last question. What is the mechanism of mRNA trafficking? In previous study, we found that some mRNAs with specific sequence are more likely to be transported. To discover specific sequence, we generate a construct containing GFP, fuse the two different fragments of mRNA, and transform it into the tobacco. We use this transgenic tobacco as a rootstock and use the wild type as a siren. We found that a fragment which was about 500 BP. It can move through can move from the rootstock to the siren. Its mobile efficiency is about 60%. And this PCR analysis revealed that all fragment will can be or not can be moved through the film. Now, all fragments were analyzed using the MME program. We found that all the potential mobile fragments contain CU-CU motif, while the fragment without CU-CU motif show no mobility. It is believed that plant PTB proteins could recognize CU-CU domains and bind mobile RNAs to form RNP complex, which plays a circular role in mediating long-distance movement of foam mRNA. Now we identify 3 PTB proteins through in dually form, and they had high homology to the 3 PTBs of Arabidopsis, and the PTB3 had the highest similarly to the CMRBP50, which is a key component of the RNP complex in pumpkin. We found that the PTB3 maybe is a potential protein to interact with the mRNAs contain CU-CU motif. So we use the RNA protein put down and EMSA experiment to detect it. We found that the PBWOST1 means the mobile mRNAs. Its whole sequence can interact with PTB3, and the fragment contain CU motif can interact with it, but the sequence without CU motif cannot. Also, for the EMSA experiment, we divided the fragment for 5 parts. 1, 2, 3, it contains CU motif, and the 4th and 5th without CU motif. The same, we found that the 1, 2, 3 fragment can interact with PTB3, and the last one cannot. So the result shows that the PTB3 can bind to CU-CU of PBWOST1 to form RNP complex. To confirm that PTB3 could assist the long-distance trafficking of mRNA in form, we generated the double transformation lines in which GFP filled to the PBWOST1, and the maturity tag was filled to the PTB3. This two construct was co-expression in the tobacco. We use this co-expression tobacco as a rootstock, and we set these groups for WT for the cell, and PTB3 only expression for the cell. And we found that the PTB3 can move through the form from the rootstock to the cell. And also we found that the PBWOST1 its relative level in the group we use the PTB3 as a sign. It has the highest level, so it means that the PTB3 can assist the PBWOST1 for long distance trafficking. And the previous studies showed that the PB-PDB3 with the mRNAs to form the RNP complex, it is not only one, but also have any other components. So we use the PB-PDB3 as a bit to screen the dually formed cDNA library. And we found about 65 clones in the screen result. It all belongs to about this biological process. Because we want to find the PB-PDB3 interaction protein, so we focus on the binding group. Now here, clones appear to repeat. It's T complex protein 1. Subnets explain. We focus on it. We analyze the conserved domain of long length of number 63, and we found that the protein contained a championing conserved domain. Meanwhile, NCBI sequence alignment showed that it had above 70% similarly with Arabidopsis and BOM and human and Eastus CCT5 genes. Study on CCT, it is a championing. In other species, it has shown that it is cytoplasmic championing in equals consisting of 8 paralogous subnets from CCT1 to CCT8. We compared each subnet of Arabidopsis CCT with the public available pair genomic sequence to identify the pair homologs, and then cloned them from dually form. Sequence alignment analysis showed that all 8 subnets contain domains that are conserved and correct in Japanism. And we found that the CCT in our dually form can classify to the cooperating subnets to the Arabidopsis, human, and the boys and the East. So we named them from PBCCT1 to PBCCT8. The 8 CCT subnets are homologs, but they are highly divergent in their apical domains. So it confers substrate binding especially. To identify the PBCCT to interact with PB-PDB3, which subnets was the interaction subnets. So we use the East2 hybrid and the BFC analysis. The results show that the subnets 5, 6, and 8 can interact with PB-PDB3. Furthermore, we divide the PB-PDB3 into 4 fragments according to its RNA recognition motif. And we found that the motif 1 can interact with the subnets 5 and 6. The fragment 3 can interact with only PBCCT5, and the fragment 4 can interact with all the 3 subnets, but the second motif cannot interact with the PBCCT each subnets. So we found that the PB-PTB3 interacted with PBCCT complex according to the RM1, 3, and 4. And in other species show that the PBCCT subnets alignment with different sets. And we want to know how about it in the pairs. So we use the East2 hybrid, the BFC, and the CoIP experiment to identify it. And we show that the PBCCT subnets, its clockwise is subnets 1, 4, 2, 5, 7, 8, 6, and 3. And the alignment of the 2 trunk of ring is here. Now we have found that PBPTB3 can interact with PBCCT complex. Well, how about its function? In previous studies showed that PBCCT, it can fold the subunits in the center of it. So we use the E. coli system to purify PBPTB3 and PBCCT1 to 8 subunits. And we incubate PBPTB3 with CCT1 or 1, 2, and so on together for about 30 minutes. And then we digested the reaction system with the protein key to